Hi everyone, so in this video I'm going to talk about G-coupled protein receptors. So the G-coupled protein receptors are actually so important when it comes to the drugs. So it's really important for pharmacology because most of the drugs are actually working in these specific manner where a specific ligand or a specific molecule in the drug would actually bind to the G protein, leading to the change of the transcription at the end. And we're gonna know this from this video here. So the G coupled protein receptor spans the cell membrane 7 times, and attached to it from the internal side is 3 subunits, and these subunits are alpha subunit, beta subunit, and gamma subunit. Okay, so in this video we're gonna talk only about the alpha subunit because it has 3 different pathways. So for the alpha subunit, we're having G alpha S, and we're having G alpha I, and lastly we're having G alpha Q. Each one of these are actually following a different pathway. For G alpha S, the first thing it does, so let's go over the general steps. So the general steps, what's gonna happen is that a ligand will actually come and bind to the protein and activate it. Let's say, for example, epinephrine in this case, and it will come and bind, making this subunits to be activated. So what's gonna happen is that these alpha subunits would actually dissociate from the beta and gamma. So this is gonna be our product here. So this is the alpha here, and it will be attached to a GTP. So this is the active form of it. Beta and gamma will just bind together away from the alpha subunit. And then after the dissociation, it could actually follow one of these pathways. Okay, so a ligand comes, and then it binds to the G coupled protein receptor, and then this will lead to the binding of alpha subunit with GTP here, and eventually alpha subunit will dissociate from beta and gamma subunits. So for G alpha S pathway here, what's gonna happen is that the G alpha S will go and bind with, so as I said, it's right now G alpha here bound to GTP. This is the activated form of it. So it goes and activate. So it will activate adenyl cyclase. And adenyl cyclase will use ATP converted to ADP to form cyclic AMP. And cyclic AMP is a very important second messenger. It's considered to be a second messenger because it will keep cascading. It will keep stimulating another cascades in order to reach to the final product. So the cyclic AMP would keep would actually activate PKA, which is protein kinase A. And protein kinase A, what it does, it phosphorylates. So it phosphorylates other cascades or other molecules until it reaches to the nucleus. So it enters the nucleus at the end, and what happens is that it binds to a regulatory sequence on the DNA, and what will happen is that it will stimulate transcription in this part. So the gene will be expressed and translation will occur, producing protein that is really necessary for this particular situation. So this is for the GS pathway. For the GI pathway, I here is for inhibition. So what it does, it only inhibit the activation of adenyl cyclase. Okay, so this is what it does. This is all what it does. For the GQ pathway, it's kind of similar to the GS pathway, but it's a little different. So here in the GQ pathway, it will follow the same initial steps. So a ligand will bind to the G protein, and then it will activate it, going to activate the alpha subunit. So alpha subunit here with GTP. So GTP is bound to it, kind of like the same. But here, instead of going and activating adenyl cyclase, it will activate phospholipase C. And it's also called PLC. And what this enzyme do, it actually breaks down phospholipids. And in this case, it will break down PIP2, so PIP2. It will be cleaved into 2 molecules, which are really necessary. And they are kind of considered as a second messenger because they have really important cascades that will lead to the alteration of transcription at the end. So it will give us IP3 and it will give us DAG. So what IP3 is responsible for, it goes and bind to the membrane of the rough endoplasmic reticulum. The smooth endoplasmic reticulum. So it binds to a receptor on the smooth endoplasmic reticulum. And as we know, the smooth endoplasmic reticulum has a high concentration of calcium. So this would lead to the openness of the channels of calcium, and calcium will efflux from the endoplasmic reticulum, goes to the cytoplasm, leading to changing the cascade. And as we know, calcium is a second messenger, and it will keep enhancing another cascades until it alters the transcription or the gene expression. For DAG, it's kind of the same thing. So for DAG, it will activate PKC. And this is a protein kinase. Also, it phosphorylates, and it phosphorylates a lot of proteins, and then it will end up going inside of the nucleus, and also alteration of the gene expression.